we didn't get into genetic entropy, but that's another whole other bag of.
Okay, so
Athena,
before we
I'm sure you're quite familiar with genetic entropy and junk DNA after what 5 hours of conversation with CRISPR.
I'll be honest with you, I was tuning out.
Well, you should tune in. So I heard you guys actually, Kevin, are you an evolutionist or an agnostic, atheistic evolutionist? Like, what do you consider yourself, brother?
I was originally raised Catholic, but honestly, I don't subscribe to any religion. I believe that if you believe in something, I think that's great if it gets you through your day, or if that's what you choose to do, if that's how you were raised. But I've seen too many things that didn't sit right with me in the world of religion for me to be able to feel comfortable to continue in that lifestyle. However, I do respect other people's beliefs. So on that note, if I may, I understand that you don't like swear words. I would just like to start out by asking you what words. I know the big ones are obviously not good, but are there any ones we might view small that you may not like? For example, hell and heck. Like, what is your stance on that?
Well, I guess the word hell is a biblical word, so I guess that it's maybe based on the context that it's being used. And I think when it comes to the swearing, I just think if we're having a discussion, like a casual talk, then based on the articulation of whatever point we're trying to make, I think it just comes across a little bit more maybe respectful and intelligent when the swearing is not added into it. And then I also usually upload these chats because they usually end up being quite engaging on Athena's channel. And then my channel is more Christian based, right? So I just would prefer there not to be swearing. But yeah, I'm sure hell or those words are fine. And I think you understand, like you said, the big ones, those are probably the ones to better avoid, right?
Right. Well, I kind of figured that. So, I mean, I do in casual conversation outside of a venue like this, I do have a tendency to use those types of words, but in the as it pertains to this particular juncture, I will do my best to not use that type of language so that it doesn't complicate what it is that you're trying to do, and so that everybody can be comfortable in conversation.
No problem, no problem. And then, so you said that you grew up Catholic, but now you're um, obviously more either agnostic or like in regards to say the origin of life, the origin of species, the origin of life, say obviously, lifeless chemicals evolving into say a single cell. Obviously, that's abiogenesis. But when it comes to the origin of species and the explanation for the diversity that we see, would you say that you hold to universal common ancestry?
Can you explain what you mean by that? I
So
know that might seem a little bit elementary, but I just want to make sure that we're understanding each other.
Yeah,
I
hang on.
think the same thing that like Athena would. Actually, Athena might be able to explain it well, but obviously, that we all descend from a single common ancestor, right? We all, the tree of life, right? Descent with modification, Darwin's theory of evolution, what I call, bacteria to biologist evolution. Like, because I would say there's limits to the change, right? Like, obviously, a domestic dog, a wolf, a coyote, a dingo. Like I would say they all share a common ancestor, but I wouldn't say that a dog, a wolf, and say a banana plant share a common ancestor, right? Like I believe there's a barrier, built in.
Well,
So that would be. Go ahead.
That's that's how he explains it. But based on what I've what I've observed and picked up over these discussions, that we all of us share a common ancestor. You share a common ancestor with hold on, Kevin. What fruits do you have in your house?
An apple.
Well, it's an honest question. I don't know. I don't know. I haven't been to his house in a week.
Are you saying that humans, banana plants, apples, and pears are all related through common ancestry, Athena?
Yeah.
Okay, so I think I asked you last time, but obviously I just stated my view that I believe there are limits, right? There's a built-in barrier somewhere. I don't know if I can exactly identify it, but do you believe, given enough time, given enough processes, and maybe Kevin can answer this too. Do you believe you can get, say, a dog as small as a mouse? Because we know they're both mammals. We know animals do exist that are as
I
small as
mean,
a
have you
mouse, like
met
a mouse. Do you believe
chihuahuas?
that's possible?
Have you met chihuahuas? And Kevin can attest to this. He once had a dog that was so goddamn tiny. Is that fine? Goddamn, because that slipped. But
I think
I,
that's
I'd
pretty
say
blas.
that's one of the bigger ones.
Yeah, because
well,
that's
sorry.
blasphemous, so you can't use
Hold
that.
on,
But
hold on. I'd like to let me explain something. Because god damn, it's not really blasphemous if you think about it. Because god damn, it's like God is damning something. So it's
Well,
not
I
really.
guess I guess I guess it's the context, but that's fine. Yeah. So you were explaining that you do believe that you can get a dog as small as a mouse, or you're saying that you can get a dog obviously as small as a Chihuahua. I've seen Chihuahuas. I have friends that own Chihuahuas. They're definitely not as small
Well,
as a
Kevin,
mouse, and they're
how
definitely not as
small?
small as a flea because we do know there's animals as small, well, insects as small as a flea, or like a flea, right? Go ahead. You
Yes,
and
but
Kevin can both answer if you'd like.
how
well, let
small?
him, I'll let him go first, and then I'll give my answer after he's done.
Oh, no. Okay, I was asking you how small was that dog? You remember the very tiny one?
You're
I forget.
talking about the one that I had.
Yes, the one that you had. Like it was so tiny. It was like, dude, you just by stepping on it, you kill it.
He was pretty small, but he was definitely, if we're gonna go mouse size, I mean, he was definitely bigger than a mouse. I mean, although I have recently on Google, I've seen that there are breeds of rats that are very large, but when you say as small as a mouse, I'm assuming that you mean like a standard mouse that you would feed to a snake.
Yeah, like I'm using a mouse as an example because we know that a dog and a mouse are both mammals, right? But I mean, we can even go as far as, can you get a dog as small as a flea? Because we know there's insects as small as a flea, like a flea. And you would say, I believe that,
I mean,
dogs and fleas are related through common ancestry, if I'm, um, you know, representing you correctly. So do you believe that there are limits, whether they're physical, whether they're genomic? Do you believe there's any limits at all? Like I know CRISPR said that there's soft limits, but how do you feel?
I would say that I don't think it would be normal for a dog to be that size. It would have to be done through specific breeding because even an animal like a dog like a Chihuahua, I don't think is a normal occurrence in the dog community that it was bred to be a size like that. So I don't think that it would be normal for a dog to be, say, the
you could,
size of a mouse.
you could probably genetically engineer a dog the size of a mouse.
You think we can
That's
artificially select a dog as, because obviously we've been breeding dogs for hundreds of years, and obviously all we've bred is a variation of dogs, and we've seen based on adaptive degeneration. I mean, we've seen a wolf to a Chihuahua. That's obviously the sorting out and degeneration of the information in that animal. So I guess my next question would be to both of you: do you believe that a dog population today ultimately came from something that was, say, non-dog long ago and far away?
I would say that I think originally, at least from what I understand, dogs came from wolves that were domesticated in one way or another.
Do you believe that a dog and a wolf, because obviously we know they can interbreed. I mean, I think anybody would say that they're both dogs, but would you say that, say, a wolf or,
Hold on,
a
they're both
dog came
canines.
from something that was recognizably a non-dog in the past at some point?
Oh, I really hate Hovind.
Like, I don't understand. Give me an example of
So,
what you mean.
so I guess I think Athena understands, because Athena just said that obviously, humans, dogs, elephants, pine trees, and apples and banana plants are all related through common ancestry. So I'm just trying to understand, because I'm saying that I believe there's limits. You know what I mean? I don't think we're ever going to get a dog as small as a flea. I don't think we're ever going to get a dog as big as an elephant. I believe there's a built-in barrier and limit there. I believe dogs will always produce dogs, but from my understanding, and correct me if I'm wrong, I don't like straw manning anyone's position. From what I understand, the evolutionist believes that a population of dogs today came from ultimately something that was non-dog long ago and far away, right? At some point, there were no dogs. Millions of years later, there are dogs. Can you elaborate, Athena?
Well, from like I said, from what I understand, both wolves and dogs or canines, like they both came from like wolf-like ancestors. You know what I mean? At one point they probably they diverged, just like humans diverged from other primates.
Well, I guess my point is it's okay to believe, obviously, that say dogs today came from something that was non-dog. But do you believe that it's empirical science? Obviously, science means to know, right? How do we know? We test, we observe, we demonstrate, we retest, we investigate. Do you believe that the hypotheses that we all share a common ancestor, and that dogs and fleas are related? Like, do you believe that that's empirical science? Because I don't believe that obviously in the present we see that, because we obviously see dogs producing dogs, but to say that dogs today came from non-dogs long ago and far away, like that doesn't seem like science to me. Do you believe that that's science or more faith-based? Kevin, and then I guess Athena.
Well, I mean, if you're gonna bring, I think that certain breeds would have to come from science in the sense that you have to do certain things to get certain dog breeds. I mean, just because it's not normal for because a dog wouldn't, a dog, say a Chihuahua size, wouldn't be able to survive in the wild per se.
So you're saying there's like physical limits, right? Based on the environment, based on physical reason. But I guess my question, because I understand that that's the theory of universal common ancestry. I'm just saying, do you believe that that is something that can be considered scientific? Because obviously, based on what science means, because it's my I guess belief that that isn't part of science, right? Because if we see dogs produce dogs, but you're saying dogs came from something that was non-dog long ago and far away, like I would say that that's more of a faith-based belief. What do you think, Athena and Kevin?
I think religion. Sorry, hold on.
Well, Athena, if I, because you do believe that a non-dog population can eventually become a dog population. We obviously don't see that today, but you're saying that through natural evolutionary processes, right? Natural selection, random mutations, environmental pressures, obviously changes in the genome. You believe that that's possible, but we just don't have enough time right now to see that, right? Like with time, um, dogs, fleas, and elephants will be related through common ancestry, right? We just don't see that deep time today, right? Would that be your answer?
I don't know.
Well, because what I'm saying is That's usually the answer, is the fact that obviously because the law of monophily states that, we can never outgrow our ancestry. Right? A dog's always going to produce a dog. A bear is always going to produce a bear. But obviously these animals did come from something that was, say, non-bear, non-dog, long ago and far away. But given enough time and processes, you will eventually get a dog and a bear. But it's that time, right? It's that deep time. Am I correct in saying that?
Well, according to the evolutionists, quote unquote.
So, are you an evolutionist?
I am somebody, I don't know.
Because, for example, because I'm just saying there's limits, right? We know that, cows can jump, and we know some cows can jump higher than other cows. But if I had an hypothesis that if I gave a cow enough vitamins and minerals that I can get a cow to jump over the moon, would you say that that's kind of crazy? Or would you say that that's something that, given enough vitamins and minerals, could possibly happen? We can't see it today, but given enough of these things, is that something that's possible or kind of crazy regardless?
I mean, that's kind of a
I mean, I don't see how.
Yeah, no.
I
And Kevin, you're absolutely right. My point is, we see dogs produce dogs, fleas produce fleas, elephants produce elephants. That's what's normal. But to say that dogs, fleas, and elephants are all related through common ancestry, that's not normal. Just like giving that cow enough vitamins and minerals, I'm saying it's going to jump over the moon. No, that's not normal. We don't see that. We know that that's not true. So what I'm saying is, pond scum to people evolution, bacteria to biologist evolution, it's not really scientific. It's not part of empirical science. Now, you can believe in it all you want, but what I'm saying is that is a faith-based science fiction religion. Would you say that you agree with me or not? Because I respect anybody's belief
by the way, we
choice.
got
It's
dragged.
when they say that that is scientific that I disagree. Go ahead.
You want Dragnet to answer?
What's that?
Dragnet's in here.
Dreg, do you believe we can get a dog as big as an elephant? We know there's animals as big as an elephant, like an elephant. I mean, given enough time, processes, what do you think?
What do you mean get a dog as big as an elephant?
Well, do you believe that there's limits to the change that we see, right? Like, do you believe that a population of dogs today, ultimately based on the theory of universal common ancestry, came from something that was non-dog long ago and far away? Go ahead.
I don't know what you mean by came from non-dog. What does that mean?
Well, just because these gentlemen were saying that obviously dogs, elephants, fleas, and banana plants are all related through common ancestry. So I'm just saying that, given what we observe today, dogs producing dogs, and I understand the law of monophily, we can't outgrow our ancestry, but based on universal common descent, what did dogs today descend from ultimately? Go ahead.
So you're referring to a nested hierarchy and not what's evolutionary possible. Yeah, those are kind of two different ends of the question.
Well, I would say that the creationist model also predicts a nested hierarchy, but just invoking a nested hierarchy doesn't exactly prove that dogs, elephants, and banana plants are actually related, say,
Well,
on that phylogenetic tree.
you don't prove anything in science, for one. You demonstrate things. So
I
if we're
agree.
going to actually discern what happens in science, we need to use the correct vernacular and acknowledge what's actually in the literature.
Can you explain to the audience what the law of monophily is?
Sure, I'll just get the dictionary definition. That's fine.
Well, like, obviously we could never outgrow our ancestry, right? A eukaryote will always be a eukaryote. Elephants and pine trees are related through common ancestry because they're both eukaryotes, right? A eukaryote will never not be a eukaryote. A dog will never not be a dog. A mammal will not ever be a mammal. But at some point, there must have been some big change, right? Because at some point, there was something that was non-mammal, and eventually we had mammals. So I'm just asking you a simple question. Do you believe there are limits to the change?
It doesn't matter what I personally believe. You do know that that's an argument from incredulity, right?
Well, I'm saying that based on the theory of universal common ancestry.
You gonna answer my question?
Well, I think no. I understand what you're saying. It's what science can demonstrate, because science means to
Okay,
know.
so we do know that the question you just asked was directly a argument from incredulity. Are we acknowledging that?
I don't think I agree with that for this reason.
Okay.
Because if I were to say, do you believe that, the moon is made out of green cheese, I think we can all agree that it's not.
No, a demonstrably false statement and saying what I personally believe about this particular field being true are two different things. So what my personal beliefs have no bearing on scientific fact.
Okay, here's a better question then. Would you say that it's
But
a
before we
scientific...
move on, we do acknowledge that that is an argument from incredulity, correct?
Even if I were to say that that was an argument,
It's a yes or no.
I don't think I do, because
Okay.
what you personally believe, if it's not actually scientific, but you're saying it is scientific, then why is that an argument from incredulity?
Okay, again, my personal beliefs, what I personally, as Dragnot, believe has no bearing on what is scientific fact. What part of that is confusing?
Because, well, I think you're using word semantics, but even if I were
Word semantics?
even if for sake of argument, I were to say it is an argument from incredulity, it still doesn't help you answer the questions that I'm asking
Well,
of you.
it wasn't meant to answer a question. It was meant to discern the difference between my personal beliefs and what's actually demonstrably true.
Can you tell me what you personally believe and what you believe is not what you believe, but what science says is demonstrably true?
Sure, I can tell you what's in the literature. My personal beliefs are going to align with the evidence, the preponderance of evidence in the field.
Now, okay, do you think, based on the scientific literature, that it states and demonstrates that dogs, elephants, fleas, and banana plants are related through common ancestry?
Sure, that is actually demonstrably true.
Do you believe that it is demonstrably true, based on the scientific literature, that a population of dogs today that we see today ultimately did come from something that was non-dog long ago and far away? Go ahead.
Again, I don't know what a non-dog is.
What would that creature be? Okay, so obviously we got abiogenesis, which is...
Are we talking about evolution now, or are we talking about abiogenesis? Because those are two different topics.
No, exactly. That's why I
well, then why are you asking me about about evidence in one field and you're asking me about another?
Well, because I'm trying to make my point here, because abiogenesis is separate from evolution.
Yeah, so then why are you asking me about evidence in one field for the topic that we're talking about here, which is evolution?
Now, the word evolution is a slippery term, because what I'm saying is, if you mean by evolution,
not slippery. No,
dogs and wolves are related.
You're not answering my question. Why are you asking me about abiogenesis if we're talking about evolution?
I never actually connected abiogenesis with evolution.
Okay, so then why is abiogenesis being brought up in this conversation then, if you're not making a connection to it?
Because you're wondering what a non-dog is, and I'm trying to say that based on abiogenesis, at one point, this is before evolution, because my definition of evolution is a change in allele frequency or gene pool in populations over time. Would you say that that's your definition of evolution as well?
I don't have a personal definition for that. It is the definition of what is purported in the field.
So based on the scientific literature and based on the textbook definition of evolution, would you say that that was an accurate definition that I provided? Change in allele frequency in populations over time?
Yes, what you provided is an accurate depiction.
Now the question is, are there limits to that change? And that's what this whole line of questioning is. You don't know what a non-dog is. I'm trying to say that based on the acronym FARM, fish, amphibian, reptile, mammal, at some point there was recognizably something that was non-dog that had to evolve through evolutionary processes, natural selection and mutation, into what we know today as a non-dog. Are you disagreeing with that?
Are you referring to, I still know my mouse is a non-dog, and by that definition. So are you talking about wolves?
I would say that a wolf, a domestic dog, a coyote, a dingo are all related through common ancestry, but I believe that there's limits. There's a barrier to that change. But I'm
Is it?
asking you if you believe that there is a limit and a barrier to that change. Do you believe that dogs and mice are related through common ancestry? Because that's where I say there's a limit. I don't believe that.
I'm not sure what you mean by there being a limitation and what that has to do with common ancestry. I'm honestly not seeing the connection between the two.
Well, when we look at the acronym FARM, fish, amphibian, reptile,
Where are we?
mammal,
Hold on,
that's
where are
going
we
to
getting
take.
that from? Okay, that actually I meant to ask you where does farm come from? I've literally never heard of that.
Well, if we look at the theory of universal common descent, obviously I'm not saying abiogenesis is part of that, but that would say that non-living chemicals evolved into that single-celled organism into a multi-celled organism, into a fish, into an amphibian, reptile, mammal, bird, monkey, human, and so on. That's what the theory states.
That's
Do you
no,
disagree with
that's
that?
actually not an accurate depiction of what happened. But is that what your understanding is?
No, that is actually the understanding. What I'm saying is to go from that and to see those changes will take phenomenal increases and expansion in novel information into the genome. So if we don't see that today, how can you say that based on the scientific literature that is actually empirically verified? Go ahead.
So your statement is that we don't actually see evolution in the current day.
Well, it depends on your meaning of evolution, because like I
Well,
said,
didn't we
it's a
just
slow
define that?
return.
Didn't we literally just define that?
Well, are you talking about change in allele frequency in
Well,
populations over time? Or are you
I
talking about
literally could have
fish?
sworn you and I just sussed out what the definition was, and then you asked me if that was acceptable. So now why are we questioning the definition that we both agreed on?
No, we agree that that's the definition of evolution. We see
Okay,
change.
so
We
if
see
we agreed
variation.
that, then why are we challenging it now?
Because if you're going to say that fish, monkeys, and banana plants are related through common ancestry, then that's not the type of evolution that I'm referring to. Because a
Oh,
change in allele frequency in populations over time does not mean that a fish and a banana plant are related through common ancestry.
We can extrapolate the way that populations change, and we can apply those models to genomes. And if the extrapolation of that model leads to the conclusion that we're talking about, it would be a justification because we know empirically that these things work.
Okay. If we look at
can you name a human? Wait, I have a question. Can you name a human that has ever witnessed a full revolution of Pluto around the sun?
just one sec. I'm not talking to Dragon. Is this Sam?
Yeah.
Yes, Sam.
Okay, what was your question, Sam? So are we ending with
Can you name a human that has ever witnessed Pluto revolve around the sun?
Can I name a human that has ever witnessed that?
Yes.
No.
Do you believe that Pluto goes around the sun? Because humans have never. Because I'm just gonna spoil this for you. We've only known about Pluto for, I believe, 80 years, coming up on 90 years soon, and it takes about 250 years for it to revolve around the sun. So how can I justifiably say that it revolves around the sun if I've never seen it?
Are you
Or maybe
saying
you
that
believe that.
Are you saying that through observations, through
Well, answer the question. I mean, do you believe that Pluto revolves around the sun, and if so, why?
we can
It
see
sounds like we need to
we can
get
see
Neil
Pluto
deGrasse.
change. No, wait, we can see Pluto change from one location to another. So we see
But we never see it revolve around the sun. So why do you believe it revolves around the sun?
Are you saying through observable calculations, we don't actually see Pluto change from one location to another, whether it's different timestamps, whether it's different observations through the telescope, through pictures? I mean, I think that's something pretty easily identifiable and
well, it's pretty easy to identify the genomic changes that happen in populations, but you deny that readily.
But I'm saying that if we look at these genomic changes in populations, are there limits to the change? And if we see adaptive degenerative changes, how is that going to take
don't change
your
the
fish to
subject.
fisherman?
How do you determine a valid don't change the subject here. Like, I know that you
Well,
like to do that.
I'm not changing the subject. I'm just saying.
Did I interrupt you?
Well,
that's
because
a
I'm
no.
not changing
Okay,
the subject.
when I interrupt you, you're free to call me out on this. But here's the thing: is how do you determine what's a valid extrapolation and what's an invalid extrapolation? Because you made the argument that we just don't see the scale of evolution, but I can point to things that we all believe are the case, that are extrapolations, and that those extrapolations work. So I'm kind of confused here. Like, are we going to argue the merits of what's in the literature?
I would say, based on the literature, do you believe that, and I think I asked this before, does the literature demonstrate that the majority of mutations are deleterious to an extent? Like, would you say
Why are
that?
you changing the subject? I mean, what determines in your head what a valid extrapolation is?
Well, I don't believe that what I stated in regards to Pluto changing from one location to another can actually be compared to, say, macroevolution. The idea that
I don't. I'm going to spoil this for you. I don't care what you think regarding that. I'm asking you I mean, that was just an analogy. Like, I trust that you understand the difference between literal and an analogy, right?
Well, isn't extrapolation millions of years ago? Isn't that different from an extrapolation hundreds of years ago? I mean, it's just too far apart. So because extrapolation millions of years ago is, say, different.
On what basis is that not valid? I mean, we use it for radiometric dating all the time. We know, for example, we know the half-life of uranium. None of us have ever seen uranium actually half-life beyond, an atom at a time. I almost said molecule, but that'd be redundant. But my point being, so, like, what on the merits of the extrapolation? Like, how do you judge an extrapolation, or is this just sort of some ad hoc argument you're using for evolution?
Well, what I'm saying, Sam, and let me finish and flesh this out, because if we actually look at the changes that you're talking about today on a genomic level, I'm not just talking about small functional changes, but radical ones, right? Because some organism would have had to learn how to, say, convert sugars to energy. Another would have had to learn how to take sunlight and turn it into sugars. Another had to learn how to take light and turn it into a interpretable image in the brain. These are not just simple things, Sam, but amazing processes that obviously involve multiple steps and functions that they would involve ultra complex pathways.
Well, I agree. Biology is cool, but what does this have to do with answering the question of how you determine what a valid extrapolation is? Like, I've asked you this question about 4 times now, and it's you go on some random soliloquy about an unrelated subject.
The valid extrapolation, if we're looking on a genomic level.
No, not genomes. I'm asking in general. Let's be honest here. Like, we all believe that extrapolations work, right? Like, I'm sure that you can estimate your time driving to work based upon where you are on the road, how fast you're going, the traffic conditions, etc. Right?
Correct,
like, we both believe that extrapolations work. Like, stop referencing biology and start talking about the actual, like, issue here, which is that, like, you arbitrarily choose which extrapolations you accept, because, like, you can't provide me with an answer as to how you determine what a valid extrapolation is.
Well, what I would say is the word science. If we're going to extrapolate something based on the evidence, well, we're going to have to do investigations. We're going to have to do observations, demonstrations, retesting. So what I'm saying, and based on everything that I've stated in the first half an hour with these guys, which you guys believe in regards to universal common ancestry, is not a valid extrapolation based on the empirical data, and I think that's evident.
I'm not talking about it. I mean, I don't know why you keep mentioning this specific. Like, can I request that you, like, actually answer the question? Because you just keep reiterating your conclusion that you don't believe evolution is a valid extrapolation. or common descent, excuse me, is a
Well,
valid
it depends on,
extrapolation.
yeah, okay, you got
Like
it. Yeah. Depends on your definition of evolution.
I get that you continue to I get that you continue to say that, but you don't actually answer the question I'm asking. So like I'm not quite sure what to think here. Like I'm asking a very clear question, which is like, when you look at data, how do you determine whether an extrapolation is valid or not? I mean, I can tell you how I do it, but I think that that's not really the thing that's at question here.
Well, I think I just answered it based on observations, demonstrations, testings, retestings, investigations. That's how we look at the evidence and the data. We're just talking about extrapolations in general here. We're not looking
So what
at a
is your
specific
problem with,
biological example.
Well, you keep, I mean, let's be clear here. You're the one who keeps bringing up specifics. But here's the thing, right? Like people who actually do the genomics on, you know, how genomes change and understanding that, their models of DNA evolution seem to disagree with what you're saying.
Well, obviously I agree in change. All I'm saying here, I think this has been the crux of our discussion since the beginning, was does that change? Because if we agree that evolution means a change in allele frequency or gene pool in populations over, say, generations, does that change have limits? Because you already know my model. You know I don't believe that God created, you know, static species, therefore he didn't create static genomes. Obviously we're going to see changes based on, you know, an unscrambling, a deciphering of this pre-existing information. So I'm not opposed to change. I'm just saying that the change appears empirically to have a barrier and a limit to it. And for someone who doesn't believe that there is, say, a limit other than, say, soft limits, the burden of proof would be on them to demonstrate sufficiently how, say, that novel, meaningful information can be added to the genome, not just reshuffling of existing information, but actually adding the novel information. Because I'm saying, and correct me if I'm wrong, I'm saying most.
So I'm sorry, you're going on for a really long time. Is there, you know, a central question that you're asking? Because you've asked like 2 or 3 by my count. I'm not trying to be rude here, but like it's hard. You give me like a list of essay questions and you expect something succinct.
Well, would you say that a valid extrapolation is accurate if it's closer in time to, say, our present time, as compared to if it's millions of years ago? Obviously, it becomes less accurate.
I don't think it has anything to do with time scale.
You don't believe that the change
Well, no, I mean, look,
in biological...
I mean, look, if it was invalid, right? Do you believe that economic predictions are all the time? Because like forecasts for 6 months are often like horrible.
But I'm saying okay.
But no, I mean,
let me ask you, because I never got to ask you this.
You get the point that I'm making, though. It's about the quality of the extrapolation. It's not about the time period, right? For the same model. Like if you're asking me internally to one model, because you're being kind of vague with the question, in terms of one model, yeah, over shorter time scales, it's going to be more accurate. But I mean, that varies from model to model. I would never ask you about, you know, an economic model that predicts stuff from, you know, 6,000 years ago, because economic systems have changed so much.
Oh yeah, no. At the end of the day, I totally agree with you. It's about the quality of the extrapolation based on the empirical data that we're looking for. But I didn't get to ask you this last time, and obviously this is your field. Like, are the majority of mutations? Because would you agree that mutations are the engine of, say, evolution? And
I mean,
after you ask that, would you say that the majority of them?
Like, dude, you can't ask a question and then go on. You can't ask a question, pause, and then go on.
Well, it's just because it was kind of a 2-part question. Okay, go ahead.
Okay, well, I mean, let's answer one part at a time. So, like, it's one mutation is one of the drivers of evolution. I mean, it's part of what can introduce more genetic variety within a population.
Correct. But are the majority of, say, mutations are they deleterious to an extent?
They are neutral or near neutral.
What would you say the difference between near neutral and then just neutral is?
I mean, anything that has an appreciable effect on reproduction or survival.
Would you agree, though, that by its very existence, a nucleotide cannot be neutral? Because obviously nucleotides would take up space. They'd affect spacing between other nucleotide sites. They'd affect regional nucleotide composition, DNA folding, and so on. So that means even if a nucleotide, Sam, say contain absolutely no information, it's still not neutral, because what would it do? It would slow cell replication and still waste energy. So much like rust on a car, wouldn't any mutation still be deleterious to an extent?
No. I mean, there are plenty
not?
of, because there are many degenerate sites in the genome where if you mutate the sequence, it makes no difference. And that includes in functional sequences. So like, I don't know what to tell you. I mean, it's not going to affect the metabolic cost of replicating the genome, because like you don't necessarily change the number of base pairs. You might even delete base pairs that don't affect survival. Like, you do realize that affecting the phenotype and affecting survival and reproduction are two different things.
Yes, well, I'm saying that based on
you do acknowledge that that's two different things, because you made that conflation. You seem to think that anything that costs anything to the cell will affect survival and reproduction, and we know that's not the case.
No, I obviously I agree that. Yeah, I agree.
Well, then why'd you make that
I think it's a moot point to what I'm saying, because obviously if these near neutral mutations are invisible to natural selection, Sam, how does natural selection recognize them and eliminate them from the population? Go ahead.
Do you not know how natural selection works?
Natural selection, I believe, is a selection process. It's a fine-tuning process. So obviously it's going to
telling me that's something. I mean, thank you for telling me something that has the word selection and it involves selection. Right?
Well, that's why I said fine-tuning process, because
But like, natural selection works by certain, differential reproduction and survival. Organisms that, breed, their genes become more common in the next generation. So obviously, if a mutation affected those odds, it would be non-neutral.
Okay, but and let me go here. If selection that we're talking about here, if it's only going to say remove the worst deleterious mutations, but then amplify only the best beneficial mutations, how's that going to solve the mutation accumulation problem? Because obviously this means that the accumulated damage would be largely invisible. Like I've said before, like rust on a car.
What damage?
Sam, remember you said you're not going to interrupt. I won't interrupt you. I'll give you as much time. But obviously, I don't agree with adaptations, but they're going to tend to be highly invisible. Say, for example, antibiotic resistance. That means, Sam, even if you presented us here with 1,000 examples of, say, adaptation via beneficial point mutation or whatever mutation it is, you're still failing to address the key issue, which is actually net gain versus net loss. Go ahead.
Well, okay, that last part had nothing to do with what I said, and I don't even think you listened to me, to be honest, because I literally disagreed with you. Like, the majority of mutations are neutral with respect to molecular function and survival and reproduction. So like, I completely disagreed with you. I think that most base changes don't affect the phenotype.
I know it doesn't affect the phenotype, but I'm saying that, like rust on a car, it goes unnoticed. It's largely invisible. That's the problem. That over time, these accumulate in the genome.
What is rust in this analogy? If you just agreed with me, and my point is that most of these changes don't affect anything, then I mean, I don't know what we're talking about here.
You're saying that the vast majority of deleterious mutations will not display any clear phenotype. That's what you're saying.
No, I didn't. Right? That's not what I said. I just said the vast majority of mutations are neutral.
Yeah.
Where are you getting deleterious from?
because that's
seriously,
where
I understand.
because
Like, this does
that's
frustrate
where
me
we
about
differ.
listening.
I'm saying they're all deleterious. They cannot be just neutral. That's where, because obviously we know there's a very high rate of visible birth defects, which all appear to be deleterious. But how come there's no beneficial, say, birth
Those
anomalies?
defects aren't necessarily genetic defects. I mean, are we
Not
changing?
necessarily genetic defects, but how come we don't see any beneficial birth anomalies at all? Because if there are so many, say, beneficial mutations happening in the human population, shouldn't selection?
Why? So wait, we're changing topics again?
No, we're still on
Well,
mutation.
you just asserted that the majority of mutations are deleterious.
But you're saying that they cannot be, that the majority of them are actually neutral or near neutral, is what you said.
Yeah, I know, but I don't know why you're going. Like, how is what you're saying connected to what I'm saying? Because like you've just asserted that they're deleterious with literally no reasoning. Like your reasoning was, if I change a piece of DNA where there's not any transcription factor binding, like literally nothing, like it doesn't do anything, changing a base pair is not going to matter.
It's, yeah, but I'm saying it's those invisible, like I'm saying that not, because one single base pair still by its existence will take up space and cannot be neutral, because it's still going to slow cell replication waste. It's going to
Wait, can I
be
ask you a
negative.
question here? Because I heard your little spiel on this. Do you think that changing a base from an A to a G in an area that doesn't have any function is going to affect anything?
Who's defining? Because I understand that what you would be saying here, and correct me if I'm wrong, you're going to say that non-coding. No, I'm answering your question. You're going to say that, or are you saying that non-coding DNA or non-functional DNA is going to absorb these mutations? That's what makes them neutral?
I don't know why you're okay. Like your use of analogies is weird, but I'm saying like there are sites in the genome where if you change, and this can be in functional sequences as well,
Yeah.
you can change bases. Like there are what are called like degenerate sequences that can have a variety of mutations to them, and it has no effect on the phenotype because maybe the kinetics of the transcription factor it binds, or who knows. We know if I change one of the adenines to a guanine, that's not going to make a difference to the phenotype of the cell nor the survival of the organism.
So are you saying that, say, some codons?
I'm not talking about codons. This is just any base. There are many degenerate bases in the genome that don't affect phenotype at all.
So are you saying that the more changes and the more, let's say,
I'm not saying any.
mutations?
I mean, I'm asking you a question. Do you recognize that that's something that exists in the genome? That there's no difference if we mutate one site versus another. This can be in functional sequences because there are.
That was my question. So
motifs there.
Okay, so it can be in a functional sequence.
Yeah,
and
it
it's
can
still
be in a
not going to change
functional
anything.
sequence. Yeah, certain changes, a large portion, in fact.
And you're saying that that's neutral. That wouldn't be considered near neutral because it's deleterious. It's not deleterious in any way, is what you're saying.
Not in any way. You're literally just changing a base.
But what's the percentage of those versus, say?
Well, no, I'm not asking about percentages. Like that's a specific case. But can we agree that that's a thing that happens?
Yes, we can certainly agree that
okay,
that
is. Then why are you saying that all mutations are deleterious? Because that's clearly just a neutral one. It doesn't do anything to the genome in any case.
Well, I think because we know so little about the genome and we're learning so much more about the genome and we're seeing that it's polyfunctional, meaning it's polyconstrained. I mean, with a little more study, that actually might turn out to be very much deleterious, because if we only speak 1 to 3% of the genome, I'm saying that every change, even that probably, is deleterious to an extent.
Wait,
I can't say right now officially that it is.
Hang on, so you just said it was deleterious, and yet at the same time, we only know 1 to 3% of the genome, so we don't know enough. So how the hell did you go from we don't know enough to therefore it is deleterious.
Well, Sam here is talking about a point mutation. So I might be able to concede. I don't know. If we only speak 1 to 3% of the genome, I can't say 100% if that is deleterious to some extent, but I'm not actually referring.
Approximately how confident are you then? Because you just said, is it either we know
Or
very
you could
little
just
or
admit
we
you're
know it's
wrong
deleterious?
here.
Now, Sam's talking about a point mutation here, changing, A to a T or whatever. It may or may not be deleterious based on the fact that we only speak 1 to 3% of the genome, and everything we
What does that
are
mean?
learning about the genome.
What
Yeah,
does
what
1
the
to
hell does
3%
it?
mean? Because like that's not something I've ever seen in a peer-reviewed journal article.
Well, how much about the DNA language would you say we add? Well, what we're going to be doing here is probably going into ENCODE, and we will talk about that, but I just don't want to go on that rabbit trail just yet because, for
Well,
argument's
where'd you
sake,
get the number from? Like, don't shift the burden of proof.
all
Don't
the
ask me the
language.
question. I'm asking. I mean, come on, man. Don't be evasive. Like I'm asking you.
Okay. I'll answer your question. By language, I'm just saying how we actually determine function. So if we haven't done these significant
Function is
knockout tests on humans, how can we know how functional the genome is? That's why I'm saying we speak or understand 1 to 3% of the genome.
Where'd you get that number from?
1 to 3% of the genome?
Yeah, that's what I asked like 4 times, and I don't know why this is like difficult to answer.
I thought you were asking why I use that term, and by 1 to 3% of the genome, where I actually got that number from was based on how much we've actually tested functionally of the human genome through these knockout tests, which is none. So I would
You
say,
realize that we can determine function without using knockout experiments.
knockout experiments would be your gold standard for determining function in these, say, non-protein coding regions, especially if I'm saying that
that's not necessary.
Because the fact we don't know what, say, DNA, deoxyribonucleic acid, does in embryonic development,
We all
that
know
is
what
how I
DNA
would say we
stands
got the
for.
numbers of 1 to 3%, because that would
Where'd
play a
you get
huge
that
portion
from?
in the genome. Go ahead.
Where'd you get that number from? Because you still haven't answered it.
So you want
Where did you get? What's the citation for that?
this? Okay, the citation. I'll have to actually look that up for you. Just through research, through
Look, man, like just say I don't know right now. I'll look it up for you.
I'll be honest with you. I'm gonna look that up for you and provide that for you as well. But I think we're also playing games here because
I'm not playing games because I have no idea when you cite a number and it's supposed to mean something and then you don't back it up. You know what, Standing, I don't want the answer to this. Abad, do you see where I'm going with this? Because it's impossible to understand what he's saying if he throws out random numbers and statistics that he clearly doesn't know about.
These random numbers just simply do not have any bearing whatsoever if you can't actually back up the source out or the investigation behind it. It's just really claims.
I don't think it's just claims at all, because if we're sitting here having a discussion and I don't have thousands of citations right in front of me, and you won't ask, how come you won't answer me the question about what the percentage is?
They're asking you for one citation of a claim you made.
What's the
yeah, exactly.
What's the percentage of our genome that we've done these knockout tests? Once you tell me what the percentage is, maybe that's 1 to 3%. What's the percentage of our genome that we've done these knockout tests?
Why are we using knockout? Why would we use knockout tests to determine every function? That's only one way. Because knockout tests, I gotta tell you, they don't always work in terms of determining function, because you can fuck up a variety of things depending on what type of knockout you do.
Okay, but fine for now. But just answer my question. Yeah, I've been trying to answer your questions. What is the percent of the human genome that we have performed these gene knockout tests on, and then we can get into whether or
Why
not they're the
why
gold
does that
standard.
matter?
What's that?
Why does that matter?
Well, can you, because that's where we're going to get the 1 to 3%. Because I'm saying based on the fact that DNA plays important roles in embryonic development, and we haven't done these gene knockout tests, there's your 1 to 3%.
Well, hold on a second, because you just said a second ago that you didn't know what the exact numbers were. So how can you hold on to those numbers and say that they're correct if you're not exactly sure that that's actually what the numbers are? And granted, I might be the dumbest person in this room right now, because it seems like you guys are kind of bouncing all over the place and I'm not really sure what the hell it is you're talking about. But I mean, if I'm following along, I thought that you said that you didn't have the proof to back up that it was 1 to 3%. If I'm wrong, anybody can correct me.
No, well,
I'm
I mean,
telling you what the
reason
problem is.
like
Okay, go ahead.
Do you understand that, like, first off, like, we don't need to go into, like, how we determine function, what function is, but there are more tests other than knockouts that determine function. So why are we only talking about the percentage of knockouts? Because there are plenty of sequences we don't have to knock out and we know what they do.
So if we're, say, looking at, because we know, for example, that ENCODE didn't actually test what the DNA does on an embryonic level, I would say that based on...
What does that have to do with what I just... Okay, I'm going to ask you a question. What did I just say?
You were, for one, you don't, and I'll tell you, let me flesh it out. You're telling me that gene knockout tests aren't the gold standard and that there's other ways to test how a region in the DNA is actually functional other than knockout tests. And these are tests that we have performed, more or less what you said.
Yeah.
Okay, well, I'm saying that...
I don't know why I mean, you're throwing out random statistics as if I'm supposed to be impressed. I'm sorry.
I'm not doing it
in the lab. I'm not impressed. You just memorized a number from a creationist who doesn't have a citation. But, like, the thing here is that, like, I don't know why that's relevant here.
Well, no, it was an ENCODE
to determine, like...
Well, wait, I got to correct you on that. It was an ENCODE scientist that actually said it's going to take 100 years to actually understand the DNA given the trajectory that of discovery that's favoring genome-wide function, based on the fact that they found at least 80% of this non-coding DNA is transcribed into RNA, which I'm saying, and the ENCODE scientists think that suggests function, but I know you don't agree with the ENCODE scientists themselves. Go ahead.
So, okay, so some random scientist on the ENCODE project speculated about something. I mean, God, we don't have flying cars yet, so, like...
Isn't he the one that actually, well, part of the team that found that at least 80% of the non-coding DNA was transcribed into RNA suggesting function? Like, aren't those the ones on the front line? Like, shouldn't those be the ones that we're taking seriously and looking at?
But I mean, if it's just something he's estimating based on thoughts rather than actual calculations, that doesn't, like, these arguments from authority don't really matter. Like, okay, but we can change subjects. Like you said, that something being transcribed means it's functional. I'm writing my thesis on RNA that's transcribed and isn't functional.
Well, I know that they looked at what was biochemically active. So that would at least indicate that the DNA is not idle.
What does that mean? Biochemically active. What do you mean by that?
Are you talking about how the non-coding DNA is transcribed into RNA, suggesting function, indicating?
I mean, how does that suggest function?
Well, that would suggest function based on the fact that why would 80% of this DNA actually be transcribed into RNA? Because if it's not actually functional, because isn't that the cell and the DNA wasting unnecessary energy? I mean, help me understand that. Go ahead.
Well, again, like unnecessary energy does, I mean, that has nothing to do with survival unless it's actually something that affects survival. But transcription itself doesn't indicate function, because also it could just be at the level where those experiments are detecting noise, artifacts.
So are you saying that ENCODE's definition of functional is too broad then? Like, because if they define
I'm
function as
confused why like you're changing subjects again. Like what about a transcribed sequence means that I can immediately assume it's functional?
well,
I can give you
I'm not
stuff
saying that
I
you have to
would give
assume that
you.
it's functional. I'm just saying that you have to at least assume that it's not idle. Now we need to do further investigations.
No,
and
not
exactly
necessarily. Not necessarily.
Well, what are some things that this non-coding DNA is involved? Like, we know it's involved in regulatory function.
What does that have to? I mean, what does that have to do with the fact that, like, you say, like, oh, it? I mean, it's like saying, like, oh, this tape gun in my room is red. Like fire trucks are also red, so I should suspect it could potentially be a fire truck. Like, no, that's not how it works. Like,
Okay.
yes, there are transcribed sequences that are functional. We agree to this.
Okay, if you wiki, like if you look up ENCODE and look at a lot of the quotes by the scientists, they're the ones saying that it's obviously not idle. So are you disagreeing with the fact that they're saying it's not idle? I mean, help me understand that. Go ahead.
Well, what do you mean by not idle?
That it is probably involved in say regulatory functions. I'm not saying I know exactly, and they're not saying they know exactly what it is doing. They're just saying that there is a trajectory of discovery that's going to favor further and future revelations of functionality. So obviously they don't know exactly what it is, but they're seeing based on this transcription that it could be functional in other ways we can't understand. Go ahead.
Okay, so that basically said, because I want it to be functional, it should be functional. Like, this was something that ENCODE was this type of reasoning was something that ENCODE was roundly criticized for committing.
Yeah, I've
like,
seen some of the criticism.
We don't know how some of these sequences actually, like, some of their definitions are just plain stupid. But well, like that's a whole different thing.
So are you saying, okay, so now I guess we're not changing subject, but so are you saying that their definition then of functional is too broad?
Yeah, I mean, like they said, any ChIP-seq track would indicate function. I mean, there are plenty of places in the genome where the RNA polymerase is bound, but it's not doing anything.
Okay, so if we look at their definition of function, which is, correct me if I'm wrong, specific biochemical activity. So this doesn't necessarily.
Do you know what they mean by biochemical activity?
Well, I know that it doesn't necessarily mean that they know the function of, say, a given sequence.
No. Forget about function and all of that. Do you know what they meant by biochemical activity? Because this is really important to understanding, what their claim was.
What they meant is that it's not idle and that it's transcribed.
No.
So what did they mean by specific biochemical activity? I'm saying they just meant that it's not idle. Now, what's it doing? Okay, go ahead.
What do you mean by idle? What does idle mean?
I would say
that you're using words that don't actually appear in the ENCODE paper, and I've linked it for you.
Okay, so
why you don't
like
read
I said before, biochemical activity means that it's involved in regulation.
Okay, I know that what they were trying to find was regulatory DNA. I'm well aware of that. But what is the actual activity that they're sensing there? Because it's not just transcription.
So you're saying that
idle does not appear in the ENCODE. I mean, I'll find it right now. I have it bookmarked. Idle doesn't appear in that paper.
But we know that transcription, for the guys listening, that's just essentially copying the DNA into RNA. But they thought that was exclusive to part, and correct me if I'm wrong, to parts of the DNA that say coded for proteins. So the fact is,
no,
what's
they
that?
didn't.
I said exclusive. So obviously, are you going to tell me that obviously they expected some of the non-protein coding regions to be involved in that copying process of DNA and RNA?
Yeah.
I don't think that they understood that as much as they
You know
found.
what a promoter is?
You cut out there.
You know what a promoter is?
A promoter sequence?
Yes.
Well, I know that they understood that was involved in the non.
You know what it is? Dude, you don't have to like go on these spiels. Like, you can answer a question just simply, yes, I know what a promoter is. No, Sam, could you explain that?
Could you explain the textbook definition of promoter?
I don't know why it has to be textbook, but my understanding of what a promoter is it's the place where transcription of genes starts, and it's where proteins such as transcription factors and the RNA polymerase complex is recruited there. It's non-coding, and we've known about it since like the 70s or 80s. So
but isn't what it's promoting the regulation of proteins?
Do you get that what you said was that they expected that exclusively only coding regions would be transcribed, but we knew that promoters were transcribed before ENCODE. We've known that since the 80s.
Yeah, I said that before you asked. After you asked me what a promoter was, I said that they knew that those promoters were transcribed in the non-protein coding regions. Didn't I say that?
Yeah, but you just said that what they expected going into the ENCODE project was that only protein coding regions were transcribed. You literally just said that, so you're kind of contradicting yourself because promoters occur at the start of every gene.
Are you saying that before 2001? Like, okay, I think this will help me understand you. How much?
Why are we talking about 2001?
Well, because I'm just saying. Well, because obviously before ENCODE, didn't they believe that most of the non-protein coding regions were idle, like they weren't.
What do you mean by
doing anything?
Using that word, but it's not something that actually occurs in the literature.
Well, based on all you got to do is go look at.
What do you mean by idle? I didn't ask you imprecise questions, but you keep using these words randomly. Like, did they find more regulatory sequences, potential regulatory sequences and, during ENCODE? Yes. I don't think that was, the extent to which was somewhat unexpected, but that doesn't affect the fact that people expected to find regulatory sequences. People knew about these sequences.
Okay, so if these promoter regions are transcribed, and we're seeing that ENCODE found that the majority of DNA is transcribed into RNA, but we know junk DNA is non-coding DNA. Why, Sam, is it being transcribed into RNA? I know evolutionists will say this RNA isn't doing anything, that there is activity, but it's just not useful activity. Why is that?
Again, I don't know what you're using as a definition of activity now, but you keep changing it, but whatever. A lot of transcripts are degraded in the nucleus and do nothing. So I don't know what to tell you. It's like a lot of transcripts are useless, or we have no measurable function.
Is it being shown that this RNA is non-functional, or are you saying this to accommodate this evidence within an evolutionary and biased worldview? Because obviously, as a biblical creationist here, I would say it's inconsistent with a logical and orderly designer, obviously who I attribute to the God of the Bible, to design organisms with a significant amount of non-functional DNA. So all I'm saying is the finding by ENCODE that this junk DNA, these regions have activity and may be functional, is consistent with my worldview. Therefore, I have no reason to give up my worldview, because to be honest, I just see a lot of bias.
Listen, I can attack you with personal attacks all day. Because all you're trying to do is say that I'm biased. Like
Well,
everyone has biases. Like I don't think when it comes to me evaluating ENCODE as a scientist, I mean, shit, like their data looks like pure crap on some of the cell lines. I'll openly say that. Like
okay, so I'm
but
not
like
crazy.
that's not a. I mean, like you need to make an argument that like transcripts don't get degraded in the nucleus. I mean, that happens all the time.
But I'm not crazy when I read quotes on Wiki and quotes from ENCODE scientists that seem to be in disagreement.
I mean, you're right.
Seems like,
Do you think if I cited Wikipedia in my PhD thesis, it would get past my committee?
well, what if you were to cite quotes from ENCODE researchers? Because from what they're saying in 2001,
I would never cite a quote. Dude, have you never written a paper for a class? Like you need to cite actual peer reviewed.
I'm not a scientist like you, Sam.
I mean, before I was a scientist, I did have to write essays that required only peer-reviewed references.
So are you disagreeing that in 2001, before ENCODE became popular, that 98% of DNA
ENCODE wasn't completed until years after that?
Yeah, because are they in phase 5? I don't have it in front of me.
Well, ENCODE, the first phase didn't get proposed until the mid-2000s.
Well, the one ENCODE researcher that I know, not personally, but I've seen in many articles and papers here, he said that almost every nucleotide is associated with a function of some sort or another, and we now know where they are, what binds to them, and what their associations are. So do you disagree with him?
I've given a reason why I disagree with it.
Okay, so no, I understand you gave a reason, and that's good, and you've also written papers, and you don't, cite these ENCODE researchers, but you do disagree with a lot of these statements from these ENCODE researchers.
Does there have to be a passive-aggressive personal attack in every single one of your comments?
No, because every time I talk to CRISPR, I've had discussions with CRISPR 4 times. You guys always try and make creationists look uninformed, ill-informed based on, what we're saying about these non-protein coding regions, but we're agreeing with the ENCODE researchers.
Can I make a point? You're Canadian, right?
Correct. Yeah. How do you know?
A Canadian was once mean to me. Therefore, I should expect that you're going to be mean to me. Why are you judging me based upon CRISPR?
No, okay, yeah, no, I'll concede that.
Let's be honest. Like, you being passive aggressive is just you being a dick to me. That has nothing to do with whether, that is literally nothing to do, because I've tried to be respectful and I've tried to just ask questions and not assert things about your viewpoint.
Well,
Standing, I'm just really curious of why do you feel the need to keep throwing out quotes? I mean, the quotes from random people doesn't really hold any weight unless, hang on a second, unless you actually have the evidence to back up that quote. I mean, just somebody saying something doesn't mean anything to us. We're not exactly impressed by it.
Well, to be honest with you, if you rewind, that was actually the first quote that I've actually provided word for word. Secondly, we've spent about 20 minutes going over ENCODE, what they found, functions that are found in those regions. So I think based on a viewing of the discussion, I mean, I think the
we can speculate as to what other people think about us. It doesn't, that doesn't matter to me, honestly. Like, when I ask you a question, I'm not just asking for my own edification. Like, okay, how do you actually justify that number beyond just quoting someone? Because I can give examples of, on technical levels, why I don't accept their argument. Like, if something is produced at the level of 1 transcript per cell, we have no idea if that's anything other than spurious transcription. And that was something like 40% of the transcripts in the ENCODE analysis.
Yes, but they also didn't test for developmental functions. So obviously, if a lot of the developmental and embryonic process involves lots of, say, construction tools, lots of workers in those non-protein coding regions. There's going to be many more functions to be found in the. Do you believe
people
that?
have done work? I mean, people have done work on, the evolution of like people. This is the problem is standing is that you're quoting science from 2010, and we're in 2018. People have probed embryonic tissues to find functional areas. Now, not at the level that ENCODE did as a consortium, because for a lot of those early time points, we can only do one single cell at a time. But you actually can do one epigenetic assay and one transcriptional assay, although the transcriptional assay is pretty weak. The other one's weaker. Like, people have profiled it, and it still doesn't really bear out this like rosy picture that all of these transcripts matter so much. Like, if anything, genes are like not expressed as much in early embryo.
But I want to comment on that, but I just want to ask you if
well, what's objectionable about what I said? I mean, you clearly aren't familiar with the literature on this. I don't know. I'm not trying to be, and by the way, not trying to be mean here, but when you say people have an epigenetically profiled embryonic tissues, that's just not true.
Well,
that's true of 2011, but we're not in 2011. We're in 2018.
But what I'm saying is, I think more of the genome is involved in, I think, that short 9-month window of time that represents human embryonic development.
On what basis?
Pardon?
On what basis?
Well, if we look at it, and then let me flesh this out and see what you think. Obviously, in that window, we see that a single cell turns into a fully formed baby that does contain hundreds of cell types that must execute an unimaginable number of cellular tasks. So I'm saying, based on that, on the construction tools involved in that, I think that it's pretty evident that much of our genome would be used in those stages. You don't agree with that?
I mean, we know that like most of the chromatin is more open during the beginning of development, but I mean, that doesn't mean that it's doing much other than just being available. So I'm wondering, based on the actual science, rather than saying things like construction tools.
Oh, I'm just using that to make it more understandable for the audience.
If that's the level, I mean, if that's the level at which you understand it, that's fine.
But if we look,
like, I don't know why you would expect, like, given the fact that you're not familiar with any epigenetic research that's been done since ENCODE,
okay,
I find this to be a really difficult discussion to have, because
let me just
don't know, based upon low input ChIP-seq experiments, how much of the chromatin that's open does. I mean, they just think it's available so that once the different expression patterns take hold in the gene regulatory sequences, do their thing and proteins bind, then they get differentiated. So they're just kind of there, not really doing anything. They're just open and potential. So, like, I'm confused when you say something like that and the current science doesn't back it up.
Well, I'm saying that based on the current science, we don't actually know the step-by-step process of what DNA actually does during embryonic development. Like, do you believe, and can you tell me what it does step by step? Because that
I mean, we can't really do. I mean, we know how it acts to a similar degree that we know how any other cells do, like, to less precision for the earlier stages, because single cell sequencing experiments are noisy, but we know, like, quite a bit.
Well, would you say that the dynamic use of, say, DNA...
did you know that we could do single cell RNA-seq experiments? This is relevant, because this is from maybe the past 2 or 3 years that this has really been a field. So if you're not familiar with the literature at this point, it's going to be difficult to have a conversation.
is that relevant to discovering what that step-by-step process is in the DNA during the embryonic development? Like if you've studied it, can you tell me the step-by-step process, the construction tools being used, being utilized, whether
What do you
it's in
mean
the
by construction tool?
Well,
like I'm trying to have a biological conversation, and like you're just not having one. I mean, like we know that the basic mechanisms, like the question isn't how the basic mechanisms of DNA work. They work the same in all cell types. I mean, like
okay,
the question,
okay, I understand what you're saying. So I guess maybe this is the answer. Like, where are the regulatory tools and the manufacturing tools in embryonic development?
what is a
Where are they
manufacturing
located?
tool?
What's the
Stop
stages
using, like, I get it, you work in construction, but like I don't. It's not something I can say in a talk. So like I have to ask again, like how familiar are you with what's out there regarding this stuff?
Well, what you just said came out in the last couple of years, so it sounds like it's a study in process. So what is that step-by-step process in the DNA?
What do you mean by a step-by-step process in this context? Like you're using terms that don't appear in the literature.
Well, would you agree that the dynamic use of DNA sequence during development is very different than, say, the vast majority of DNA sequence being used in the adult?
Different in what sense?
Well, the use of the DNA sequence,
it's used the same way. So again, like I don't know what you mean by this. Like you keep using these vague terms, but what is fundamentally different about development that's different than a normal cell type in terms of the mechanisms of how, you know, the genome is regulated?
Well, I know the genome is regulated the same throughout, but the various.
So then why do you keep saying new uses?
Well, the use of it specifically on a DNA level during those embryonic stages, because I'm saying that experimentally testing, say, a DNA sequence during each of those unique windows of time we're talking about, from human embryo to baby, in those sections of DNA being used and then silenced would be enormous.
I mean, the only evidence that we have is basically that it's just a more generalized regulatory architecture that gets specified into you know, different cell types. Like there's no evidence that like I'm sorry, but the prediction of ENCODE that nearly all of the new like that they would discover even more sequences in embryonic areas. It just doesn't seem to be the case. It just seems to be that it occupies the union of that 80%.
Well, from my understanding, I could be wrong on this because I don't have a citation. Aren't there expressed RNA sequences that have been analyzed in aborted fetal tissue, and I think even in embryonic stem cells?
What does that have to do with anything I just said? Like, if you're just not familiar with anything in epigenetics,
well, no, I'm saying that the data.
When's the last time you read a peer-reviewed article from, you know, a relevant journal on this topic? Because like I feel like I'm talking really past you and you don't understand what I'm saying.
No, I understand. I feel like based on everything that I'm saying and based on the question about that, because I don't think that you can name what the DNA does step by step in development, and I think that that's a strong indication that you're admitting.
What do you mean by?
He literally just asked for when did you last read a journal on this subject, and you went on to a completely different angle.
I'm actually curious about that too.
Well, the reason why I didn't answer that question right away was because then he went on another rant about talking past me and all this.
Then can you please answer that question now?
When was the last time I read an actual? Because I know that obviously something from a Creation Ministries related journal isn't going to satisfy him.
Peer review.
Guys. The exact stipulation was a journal from a peer review publication.
Okay, that's good. So then that would have been yesterday. I can send you the paper. I don't think you're going to accept it because it's through Creation peer-reviewed journal, but that's fine.
Those aren't considered peer-reviewed journals.
They're not, as a matter of definition.
By whose standards?
By the standard that they don't conduct peer review.
So far, I haven't seen you telling me what the DNA does on a developmental and embryonic level, and I think it's pretty evident to anybody.
It does the same
listening
thing
in an unbiased point of view.
Wait. You made a specific allegation at me, so I'm going to ask. Did I? And I'm going to ask to someone who I've actually discussed stuff about, papers that do, single cell sequencing on open chromatin, Drag.
Oh,
yeah,
shit.
Discussed.
Didn't we have an entire journal club just on that?
Yeah.
I could have swore we had one. I could have swore it's still on YouTube.
And what do you know? That the open chromatin, when you look at single cell profiles, it just ends up in aggregate looking like the multi-cell, profile.
So how much? Okay, so how much would you say, based on what you know and what has been looked at in the last couple of years?
But you just said that you literally just said that I didn't talk about how it works, and I literally talked about the fact that it's open chromatin. Like I literally went on a little spiel about how it was open chromatin, where then the epigenetic regulators can begin to occupy it as it gets more and more specified.
Yes,
and I did directly say that. So then when you say something to presumably insult me, it's demonstrably wrong. So either you're just trying to insult me because you're frustrated, or you're not listening to what I'm saying.
So you're telling me based on that, that's telling me what the DNA does step by step on an embryonic
literally
level?
At the level literally at the same level that ENCODE did.
Okay, so if that's the same level that ENCODE did, but ENCODE comes to a totally different conclusion than you, who's right in the matter?
Well, the data is the same. The conclusion's different.
Yes, the conclusion's different, but if they're doing the research...
And also what they said, they speculated that they'd find more functional regions in embryonic tissue, and they haven't. That was a prediction. Like, we didn't know how development would work. We do know that more chromatin is open, and early in development, you tend to see more gene expression in general, just more genes. We know that, right? But we don't see what this bold prediction of ENCODE is empirically. And you want to know why they haven't addressed that? Because the actual methodology to do that sequencing did not exist during ENCODE. They never did single cell sequencing as part of the initial ENCODE effort. Okay, so are you going to send me the specific and appropriate papers that deal with what you're talking about? Because I want to see what they're writing and finding about the DNA process on the embryonic level. Because from my understanding, and maybe I'm just not up to date on it, they don't actually know, and there's a lot of guessing involved. But you're telling me that they do know. So I just want to make sure I'm not being tricked. And I'm not going to do your homework for you. Like it's an entire field. I can't just give you one citation. Like, it's not my fault that you're ignorant of the field. Okay, wait. But they haven't found some radically different. Wait. I just had over an hour conversation about CRISPR on this, and he never mentioned these papers. And apparently, he's a scientist. So is he ignorant on this? Different areas. We work in different areas. Do you realize that there's a lot of people that do research, and they don't all read the same articles or do the same? I'm just saying, in my exact same PhD program, and yet he is not up to date on the field of epigenetics because he doesn't research it. Okay, and all I'm saying is I want to make sure that what's being said is accurate, because from my understanding at this time, they don't know, and they haven't studied much of what the DNA does on the embryonic level. Your own admission, I'm going to look at the papers, and I'm going to see. I'm not going to do your homework for you, though. You also have to respect that, because it's honestly I'll be honest here, because the way that you talk with confidence about a subject that you don't really know anything about is kind of startling. And then you have the gall to be passive aggressive and condescend to me in the middle of your soliloquies. So like I'm kind of confused when I'm the one that, you know, does this for a living and actually deals with the data. Like, you don't even know what your main finding says. Like, you don't know what ENCODE actually assayed. And yet you're citing it. Like, your argument is just ENCODE found it. Like, you haven't found, you haven't made an argument that there's some logical connection there.
Well, I'm just going to go back into repeating things about what the non-protein coding DNA is involved in, and based on everything that I think that they've discovered, it seems like the ENCODE researchers are in agreement. There is a trajectory for genome-wide function, which would be a prediction made by creationists for probably 10 or 20 years. So if that's something that can be substantiated, that's pretty amazing. Go ahead.
What do you, but like, again, like, what did they actually do as experiments? I mean, you mentioned RNA sequencing. They did more than one kind of RNA sequencing. I believe that batch did. But do you know what the other experiments they did?
Are you saying other than looking into the specific biochemical activity that they've done in those regions?
What do they mean by biochemical activity?
Well, we're not going to go back into that. Why don't you tell me and the audience what else?
I think I'm going to. No, let's go.
I've already answered that question 3, 4,
no, you
5.
haven't, because you keep saying specific biochemical activity, but all you refer to is transcriptome, and that wouldn't account for the entire 80%.
Well, non-coding
genes. The other problem is that, like, they used other assays, and there are many transcripts that are just simple noise, or they can't map them properly.
Okay. Yes or no?
For a variety of reasons. So, like, what else did they assay?
Okay. Answer this yes or no, because I need to know what you believe is considered functional. Them finding that non-coding genes, telling proteins what to do and when to produce their product, is that function in your eyes?
Okay, I don't know why you're not answering my question, so I'm just going to refuse to answer your question and ask.
Then I'm going to refuse to answer your question. I've already answered more than enough questions from you, and if we're going to continue on this subject,
well, like, here's the thing, Standing. Do you want me to address you as a peer, as someone that I'm informing on this, or like, how do you want me to address you if we're talking about this subject? Because, like, you've admitted you're not up on the field, which means that you've really just read creationist summaries of ENCODE.
I believe that I'm up on the field, which is why I said that I'm going to look into the specific papers and work that you've provided, because I personally don't believe that what you're saying on the
how do you know you're up on the field if I literally
the developmental stage is actually accurate? I'm not saying that you're lying or misleading me or anything, but I just want to make sure, because if it's not something I've heard of, there's obviously something CRISPR didn't hear of. I'm going to look into it and see how.
Do you know what single cell ATAC-seq is? Do you know what single cell ATAC-seq is?
Single cell ATAC-seq.
ATAC-seq.
No, I don't know what that is.
Okay, then how can you say that you're up on the field? Like, if you don't, like, dude, like, I don't know why you are insisting on lying to me about this. Maybe you're lying to yourself.
You do know that you using fancy terms that you are used to during your field.
Isn't that in the paper?
Isn't you proven? Because actually, through this entire discussion, we haven't really proven at all that universal common ancestry is true. Nobody's shown that there's
great.
Let's just change the topic. It sounds like you disagree with what the ENCODE researchers have discovered. It sounds like the trajectory for genome-wide function.
Can I butt in for a second there and just calm things down and explain something for a moment?
Yeah, thanks.
It isn't Sam's fault if you're unfamiliar with the standard language of the field. If you need him to explain the terms in some other ways such that you understand it, just ask.
Yeah, but if he's referring to ATACs, because I know that if the entire genome is looked at in regards to, say, DNA binding in proteins, is that being used in the research for this, what's going on in the embryonic stages? Go ahead.
I'm just gonna change topics here because like Nicola made a great thing. Like, dude, you don't need to be as insecure as you're being right now. Like, it's honestly kind of childish.
To be honest, if I can interject there, Sam, when somebody starts bitching and whining and complaining that, well, you're just using big words to try and confuse me, I think that's actually the point where that topic is done.
I mean,
I think enough has been said on that topic.
Well,
this
Stan,
point,
can I ask a question that's actually a little bit easier? Because, like, let's be honest. Like, if you were familiar with the ENCODE science, they hadn't invented single-cell sequencing methods at that time, or at least developed them to an appreciable level. But do you know what ChIP-seq is?
Say that again. ChIP-seq?
Yes.
Define ChIP-seq. But I still want you to define ATAC-seq too. Define them both, and then tell me how they pertain.
He's asking you whether you know anything about it.
No, I don't. That's why I asked him to define it. But like I said, all of this I don't think is really proving anything. That's the whole point.
I mean, do you need me to explain what ATAC-seq is? I'll explain it. It's a way of sequencing to find open chromatin. It stands for assay for transposase accessible chromatin using sequencing. Good?
Good on that one. Now give me the definition of the other.
I'm glad I gave you time to Google, but I asked you what ChIP-seq is.
I didn't Google it because I was listening to your definition on.
No, he's already admitted that he doesn't know what ChIP-seq is.
I'm not going to be dishonest and go Google something I don't know. Everything we're talking about.
I mean, you've been dishonest elsewhere in this conversation, so I don't know why that would stop you.
Say that again?
You've been dishonest elsewhere in this conversation, so I don't know why now would be the point to stop.
Well, that might be a subjective opinion, but let me interject here. Let me interject here for a second. I think the basis of this disagreement here at the moment is the fact that, Standing, you obviously, and you've admitted as such, that you do not understand what the single cell attacks and also ChIP-seq are, and yet at the same time, previously you said that you were up to date on the literature. And that you could actually be considered a peer. You could be considered a peer. Now, obviously, I don't know this subject, but I'm assuming in this case, knowledge of those terms is kind of required to be up to date with the literature.
I've never said that I know as much as, say, Sam, who obviously works in the field, but I think I know enough to prove a point and to make a good case, say, for the biblical-based model, because I don't understand what those terms have anything to do with the relevant discussion at hand. Would be my.
Oh, I'm so glad you asked that. So, I would like to go to the ENCODE project. So it's actually worse than that because ChIP-seq was the most widely used experimental assay in the ENCODE project. So if we're going to talk about the ENCODE project, we're going to need to actually talk about the data, and the majority of experiments that were carried out were not RNA-seq, they were ChIP-seq. So by your admission, you don't know what ENCODE actually contains. Why are we discussing it as a citation?
Were these done in the latest phases of ENCODE?
They were done in the earliest phases.
And what did they discover when doing those?
You don't know what
levels of function.
The exact findings you're referencing, because do you understand that the majority of experiments that they ran in the first phase were ChIP-seq experiments?
I want to know what you're saying that they found, because if most ENCODE scientists agree with obviously what they believe about functionality, but you're saying that these other tests that they've done is, say, disproving what they hope to find or what they believe they're finding. I need to go look at both sides.
I said that that's the actual data you're referencing, and you don't know what it is. Do you see the problem there? Because I can give examples of how certain chip-seq binding probably doesn't mean anything. Is he there? Am I talking to myself?
Well, you don't like me interrupt. I'm just letting you finish what you're saying.
No, I'm asking you, like, if you don't know what this actually measures, and we want to actually talk about, because I can give a good reason for why most of the places where RNA polymerases bind don't do anything, and you don't know what you're talking about here. Like, how do we have a discussion?
Well, why don't you take a minute, present your case as to why you disagree with the ENCODE scientists, and then I'll take that and go look at the relevant papers that you've stated are relevant to the step-by-step process in the DNA in regards to the embryonic and developmental stages.
Not my job.
And I'll conclude if what you're saying is based on
not my job to do your homework, man.
or trickery. So go ahead, make your case.
I'm curious. We'll hold on. You want Sam to go and do the homework to find the data, and then you were going to gauge whether or not. Well, you asked him to find the relevant material. Did you?
No,
did you not?
I said I'm going to go find the relevant material, the relevant papers, because like I said, I've done 20 of these discussions. No one has stated what Sam has stated here, so I'm going to go look. But he wants to make his case based on the ATAC-seq, and I'm asking him to make that case so I can use what he says to go and make sure that what he stated isn't misleading. That's all I said.
I mean, I think you're severely misunderstanding what I'm saying. I'm just giving an example of what type of experimental data is there. Like, it's not my fault that you are not familiar with what the experimental data is here.
Yeah, and I was actually going to ask you about that, Standing. So if you're going to go and do research and you're not familiar with the techniques that they use to acquire the data, how are you going to gauge whether or not Sam is accurately depicting what happened?
I'll be able to, trust me on that one.
Well, if you don't know the techniques, then how are you going to be able to do that?
If you lack the prerequisite understanding to determine what is or is not relevant, how the hell are you going to determine what is relevant?
Well, I think at this point, Sam has made his case. We can move on from here. I can, like I said, look up the relevant papers. I would also
hang
like
on a
to
second. I'm still caught on back on that last point though. If you don't have the relevant training or the relevant skills or the relevant knowledge, prerequisite knowledge about these subjects, how are you going to assess anything?
So do the ENCODE scientists and researchers, do they have the appropriate and necessary and sufficient qualifications to make their own conclusions? Because like I said, if I'm going to go look at what Sam's saying...
Well, the thing is, you're not employing the ENCODE scientists to assess it. You're trying to assess it yourself.
Yeah, he's saying I'm wrong.
What you're trying
and
to
I'm going to go see. He's saying I'm wrong.
Well, why? I mean, why are we taking the ENCODE analysis as if it's gospel?
Oh, I don't think anybody's. No, I agree. I don't think anybody's analyses.
Well, then why are you defending it so ardently? Well, why are you defending their analysis so ardently when you don't understand the actual techniques that they use to generate the data?
I'm saying that from my understanding and from what I've seen, even in the last, say, 20 discussions I've done, most scientists, including ENCODE researchers and scientists, agree that there's a strong trajectory for genome-wide function, but only biased evolutionists disagree, which means I'm going to go look at what Sam's saying in regards to what we were talking about on a DNA level in the embryonic phase and see how accurate it is. I don't know how many more ways I can specify that, so we might as well change topics at this point because we've been going on this for a long time.
Yeah, but
well, no, I mean, like standing. But here's the thing: is you still keep Abad, but you still keep referencing that it's your understanding of the science, and we've exposed that you don't understand like what they did in the experiment. Like we've exposed that at this point. when you admit you don't know what the most common experimental data type that they used is, then I mean, we're sort of pissing in the wind here, because like I can give a lot of reasons as to why I don't think that their analysis would work on a technical level. Because I don't have a problem with the data. I mean, I do have a problem with the data because the data is shit compared to the techniques we have now. That's not their fault. But, you know, I'm arguing with how they interpreted and did their analysis.
You're arguing with how they did their analysis and the conclusions they made based on the techniques they used to confirm what's functional and what's not, right?
Yeah, I'm arguing with what their interpretation is.
Okay, no, I understand that. I understand that. I think that that's pretty evident to me, to you, to everyone listening in on the discussion. I'm gonna have to mute myself for 1 minute, and you guys can go ahead amongst each other. We've been going at this for a while. Just a minute.